So in this video lecture, we'll study about the ELISA. ELISA is basically a type of immunoassay. Immunoassay means that it is a method to measure any particular substance in a mixture using its specific binding antibody. One of the uh, qua- quality of immunoassay is that we can measure a substance that is present in a mixture of various contaminants. Immunoassays have been popularly in view of their high sensitivity, safety, economy, and simple instrument requirements. So, what is antigen? Antigens are basically substance that is capable of stimulating an immune response, specifically activating lymphocytes, which are the body's infection fighting WBCs. Now, what is antibody? A protective protein produced by immune system in response to the presence of foreign substances. So that is called an antibody. So this is the structure of the antibody. It is having the L chain and H chain, that is light chain and heavy chain, and antigen combining sites are there, and there are H chain, and in between the two chains there is a Hinge region. So this is the structure of the antibody. Now what is ELISA? ELISA is basically an enzyme-linked immunosorbent assay. So antigen of interest is absorbed in a plastic surface, that is sorbent. Antigen is recognized by a specific antibody that is called immune. So antibody is recognized by the secondary antibody which has attached with enzyme, that is called enzyme linked. That's why it is called enzyme linked. So substrate linked with the enzyme to produce a particular color. Usually it can produce a orange color or it can produce a blue color. So ELISA is a biochemical technique used mainly in the immunology to detect the presence of antibody or antigen in a sample. So it is a place-based method assay designed for detecting and quantifying substances such as peptides, protein, antibody, and hormones. In ELISA, an antigen must be immobilized on the solid surface. So the first requirement is it must be immobilized on the solid surface and then complex with an antibody that is linked to an enzyme. The detection is accompanied by assessing the conjugate enzyme activity via incubation with a substrate to produce a measurable product. The test can be done by polystyrene tube, macro ELISA, or polyvinyl microtiter micro ELISA. So these are two types. Now, what is the principle? ELISA is based on antigen-antibody reaction. So this is important. ELISA is based on antigen-antibody interaction, which is representing the chemical interaction between antibodies produced by beta cells of lymphocyte and antigens. The specific immune system produces an immune role in protecting the body from invaders such as pathogens and toxins. Hereby exploiting this reaction, ELISA permits the highly sensitive quantitative and qualitative analysis of the antigens. To detect the molecule, an antigen and antibody is labeled using enzymes. An antigen is the fluid phase an antigen is the fluid phase is immobilized to the solid surface, such as microtiter plates constituting polystyrene, polyvinyl chloride, and polypropylene. Subsequent, an antigen is allowed to react with the specific antibody which is detected by enzyme labeled secondary antigens. So the development of color by using a chromogenic substance indicated the presence of antigen. So antibody bind therefore ELISA can be used to detect either the presence of antigen or antibody in the sample depending upon how test is designed, as you can see in the figure. So there are different kind of ELISAs like direct ELISA and indirect ELISA. Then there is sandwiched ELISA, and then there is competitive ELISA. We'll discuss this in subsequent lecture, or we'll discuss this is in detail as we progress through this video. So steps involved in ELISA. First one is antigen binding. The sample antigen is binding or immobilized to the microplate via absorption to the surface. This is the first step. The binding is achieved by incubating the wells with a solution containing antigen for 2 hours at room temperature or overnight at 4 degrees Celsius. The protein adheres due to hydrophobic interaction between protein and plastic. Coating is done using carbonate or bicarbonate buffer at pH 9.4. The second step is blocking. All unbound sites of the solid support are blocked to prevent non-specific binding of the antibody. Blocking buffers like BSA, non-fat dry powdered milk in PBS or TBC at pH 7.4. Protein in blocking solution will be attached to membrane in plate where the target protein have not been attached. So excess blocking agent is removed by washing the plate membrane with the washing buffer, so that excess blocking can be removed. Now primary antibody. Step 3 is adding primary antibody. The primary antibody is added and will be bound only if there is any recognized epitope within the sample antigen. And next step 4 is the secondary antibody. An enzyme-linked secondary antibody is added with suitable dilution, which will bind to any available primary antibody. Secondary antibodies are linked to enzyme through biconjugate. Plate is washed with buffer or mild detergent to remove any unbound antibody or proteins. Now, fourth step is detection or development. After all final wash step, the plate is developed by adding an enzymatic chromogenic substance to give color. The entire plate is placed into plate reader, and OD is determined for each well. The intensity of the color reflects the amount of the specific secondary antibody bound to the antigen. Now, there are different types of ELISA, as I have already told that I will explain all the ELISA, like direct ELISA, indirect ELISA, competitive ELISA, and non-competitive ELISA, which is also called the sandwiched ELISA. So, in direct ELISA, in 1971, little bit history, Ingel, Perryman, Van Wieman, and Shurus were first to develop direct ELISA, which was base style for other types of ELISA. This is the base style. Primary antibody or antigen immobilized on the surface of the microtiter plate. After the surface is blocked with other protein to avoid the non-specific binding absorption of the other proteins, the corresponding enzyme like antibody or antigen is allowed to react with the immobilized target, followed by color development with appropriate substrate. While increasing the amount of target, the signal increases. Direct ELISA is suitable for the quantitative analysis of the macromolecules. So this is the figure which shows the direct ELISA. So there is first step is antigen or antibody binding to the surface. And then the second step is the incubation with the enzyme, label antigen or antibody. Wash the unbound antigen or antibody out and develop color with the substrate. So this is the direct ELISA, where first the antigen is immobilized on the surface, then the antibody is applied, which is having the enzyme, and so the antibody is enzyme labeled, and there will be a detection and formation of the color. So in direct ELISA, there is direct interaction between the antibody and antigen, and there is no other interaction takes place. So what are the advantages of direct ELISA? So advantage of direct ELISA is a quick method since one antibody is used. So this is very quick. You need not to add secondary antibody. So cross-reactivity of secondary antibody is also eliminated. So the disadvantages of direct ELISA is immunoreactivity of primary antibody may be reduced as a result of labeling. Labeling of energy primary antibody level from an experiment to another. This is the disadvantage, and little signal amplification will be there. But when you use secondary antibodies, signal amplification will be good. Now we'll see what is indirect ELISA. So development on the basis of the direct ELISA. Direct ELISA is the base. So the presence of antibodies, anti-sera, that is primary antibody. The key step of this system is the two binding process. There was only one binding process. So here is two binding process of the primary antibody and enzyme, linked secondary antibody, which is labeled with enzyme called the indirect ELISA. Antigen is primarily immobilized on the surface of microtetraplate. This is the first step, which block the surface with blocking proteins. Okay. Then primary antibody binding to the immobilized antigen is allowed to react with the enzyme-linked secondary antibody, followed by development of the color. So here only secondary antibody is the addition. That's why it is called indirect ELISA. So signal increases with increased amount of the immobilized target antigens. So this is the figure which shows the indirect ELISA. So first is the attachment of the antigen to solid surface, then incubation with primary antibody, then washed unbound antibody out, then incubation with the secondary antibody and development of color. So there will be more amplification of the signal as compared to direct ELISA. So what are the advantages of the indirect ELISA? So it is highly sensitive as compared to direct ELISA. Indirect ELISA is very sensitive. More than one label antibody is bound per antigen molecule. It is flexible also. Different primary detection antibodies can be used with a single label secondary antibody. And cost saving also. Fewer labeled antibodies are required. Now competitive ELISA. In 1973, Belanger developed competitive ELISA. The key event of the competitive ELISA is the competitive reaction between the target antigen antibody in the sample and enzyme labeled antigen antibody against corresponding immobilized antigen or antibody. To detect the antigen in an enzyme labeled antigen is used to compete with the target antigen against the immobilized antibody. Hence, the higher amount of antigen in the sample, the lower amount of the enzyme-linked antigen that binds to the antibody. This is with increasing amount of target antigen signal decreases. So advantages of the competitive ELISA, highly specific. Since two antibodies are used, the antigen analyte is specifically captured and detected. This is suitable for complex samples. The antigen does not require purification prior to measurement. Flexibility and sensitivity is high since both direct and indirect methods can be used. Sandwiched ELISA. The target antigen is detected via anchoring between two antibodies, which recognizes different epitopes. So it is called sandwiched. Sandwich ELISA starts from the immobilization of an antibody called capture antibody on the microtiter plate. Sample is added and any antigen is present bind to the captured antibody. Antigen bound to the captured body is then sandwiched with the enzyme-linked antibody for the color development. Signal increases with the increase in the amount of the antigen. So this is the figure which shows the sandwich ELISA. First is the attach captured antibody to the solid surface, then incubate with the target antigen, wash bound target out, incubate with the enzyme-linked antibody and develop the color. So here, first you attach the captured antibody to the surface, then incubate with the target antigen and incubate with the enzyme-linked antibody. So your antigen is basically sandwiched between the two antibody. So it is more sensitive as compared to direct or indirect ELISA because your antigen will be captured in between two antibodies. So what are advantages of the sandwiched ELISA? Because these are advantages like it is highly specific, suitable for complex samples, flexible and sensitivity. It is have high flexibility and sensitivity. The applications of ELISA, there are different kind. We should know applications of ELISA because when we study something, we should know applications. So basically it is involved in detection of HIV antibodies, detection of microorganisms, toxins produced by them, food allergens. It had also found application in the food industry in the detection potential food allergies, serological blood test for celiac disease. ELISA can be used as detection method for detection of mycobacterium antibodies in the tuberculosis, detection of rotavirus in the feces, detection of hepatitis B, markers in the serum, detection of HIV antibodies in the blood samples. So this is a paper, the Journal of Immunological Methods. Here they have studied an enzyme-linked immunoassay for quantification of the human colectin 11, CL11, CLK1. You can read the paper. You will get a lot of informative things here about the ELISA if you want to explore more. So these are some of the kits I have just added to show that there are a lot of commercial available kits like Invitrogen, the thermoscientific company which develops ELISA kits. So you can just take it and do your plan your experiment accordingly. Thank you. That's all for today, and hope this video helps. If you have any queries, kindly ask in the comment section, and please like and subscribe my channel. I hope this video helps, and I need your support. And thanks for being with me, and hope you understand the topic fully and